Hello everybody, today we're going to talk about the splitting of glucose. The term is glycolysis. The first part of the term here means sugar, glyco. Second part of the term, to break. Okay, so this is going to be a process that happens both in eukaryotic and prokaryotic life. Pretty much all cells, as long as they have the appropriate machinery, are going to be able to break sugar and gain some energy from it. The goal of all aspects of the breakdown of sugar is to convert the bond energy within the sugars into ATPs. Okay, so that's the overall goal, is to be able to convert them into some form of energy to be utilized on a cellular level, and ATP is the target molecule that we're looking to make. So in terms of eukaryotes or prokaryotes, as long as they have cytosol, we're going to be able to go through this process, cytosol and the appropriate enzymes. So this is going to take place here, has nothing to do with the mitochondria, okay, and it's going to take place irregardless of whether oxygen is present or not. So it has no oxygen requirement. Pretty much every cell that's alive on the planet is going to be able to break down sugar and gain some ATP from that cellular breakdown of sugar. Okay, so let's look at the specifics a little bit about how sugar enters a cell. Okay, so let's look at a eukaryotic model. We have a cell membrane here. We have two proteins that are transmembrane proteins embedded in the cell membrane. This first one here is going to be an insulin receptor, so it's going to be able to allow for the attachment of a very specific molecule to it. That molecule is insulin. It's produced by the pancreas, and when blood sugar is high, what's going to happen is insulin is going to get released into the bloodstream. It's going to attach to the insulin receptor. That is going to then allow for a signal transduction cascade to occur on the inside of the cell. The net result of that signal transduction cascade, so again, there's several iterations there of intermediates, but the end result is that a ligand is going to attach to a secondary transmembrane protein that is located somewhere else on the cell membrane. So when this ligand attaches here, what's going to end up occurring is that this protein will change shape. It's going to open up, and that's going to allow for glucose to enter into the cell. Now the one depicted here is the GLUT4 glucose transporter, but there are a couple different iterations of this kind of model that we find in eukaryotes, and when blood sugar is high, insulin is what's going to enable the sugar to enter into the cell. Okay, so once inside there, what's going to happen is we're going to break our sugar molecules into pieces, and we're going to utilize enzymes to do this. Now there's substrate level phosphorylation, where the enzyme is going to be active in splitting the sugar in half, and then if oxygen is present, we're going to be able to use all different types of activities here to break that sugar down into very small pieces. And in doing so, we're going to be able to extract way more energy out of this model than we would from this. Okay, and I hope that that makes sense. But again, enzymes are the tool that are going to utilize the splitting of the sugar. So glycolysis is this over here. We're just going to break that molecule in half. Okay, so what ends up happening is we're going to add two phosphates from ATP molecules to our 6-carbon glucose. This phosphorylation puts pressure on the molecule and is going to enable it to be split. So this requires an input of energy here, so input of 2 ATPs. Overall, once that molecule splits, we're going to be able to net 2 ATPs. So we actually get 4 out, but because of this initial cost of the 2 ATPs, we only are able to net 2 ATPs. In addition to that, we're able to take 2 pyruvates. So we took a 6-carbon glucose, we split that in half into 2 3-carbon pyruvate molecules. Those are going to be able to enter other pathways. And lastly, we're going to be able to pull some electrons off of that sugar molecule, and that's going to be stored in the form of NADH. So what happens is we have an electron acceptor, namely NAD+, and we strip electrons, we oxidize this glucose, and we stick those electrons onto this molecule. Those electrons are representing temporary energy storage, and they're going to be able to be utilized elsewhere in the cell. So again, overall products of glycolysis: take a sugar, break it in half. We got 2 pyruvates, which are each 3-carbon. We got 2 ATPs, that's net again, and we have 2 NADHs. So these are 2 electron carriers. Okay, so if there is oxygen present, we're going to go into the citric acid cycle, and then we go into electron transport chain be able to really maximize the ATP production. If there is no oxygen present, then the cell, based on the cell type, is going to enter one of two pathways, and these pathways are referred to as fermentation. So after the split in the eukaryotic cell, what's going to happen is the glucose is going to be converted to pyruvate. We already discussed that. If oxygen is present, then that pyruvate is going to get converted into acetyl-CoAs and then enter into the mitochondria. From there, we're going to have the citric acid cycle and electron transport take place inside this organ. Okay, if there is no oxygen present, then in the cytosol of the eukaryotic cell, the cell will actually convert the pyruvates, will put the electrons back into those molecules, and convert that into a lactic acid. This is what fills our muscles if we're doing aerobic activity, prolonged aerobic activity, if you're lifting weights really hard, cause your muscles to be stiff, and eventually we're going to have to pump that lactic acid out and convert that back to be able to gain some of the energy back from that lactic acid molecule. Okay, so that happens in animal cells, lactic acid fermentation. Our second type of fermentation occurs in plants, also occurs in prokaryotic cells, yeasts, but this is called alcohol fermentation. So we have glycolysis again, that happens in the cytosol. If oxygen is present, actually prokaryotic cells are capable of undergoing the Krebs cycle. That happens in the cytosol in prokaryotes. And they also have electron transport proteins embedded in their cell membrane. So if oxygen is present, prokaryotic cells are able to go through all three of these pathways and really break apart that sugar molecule, maximizing ATP production. If after glycolysis there's no oxygen present, regardless of whether we're talking about a yeast or a bacteria or a plant, what's going to happen is those pyruvate molecules are going to get broken down into alcohol. So each pyruvate actually is going to have carbon dioxide removed from it, and that's going to be released to the environment. That carbon dioxide is what's exploited when we add yeast to dough to try to make like bread rise or baking products rise. That whole gaseous release by the yeast is this carbon dioxide that's being released from the fermentation process. And the alcohol, of course, is what's exploited in winemaking or beer or any of those other kinds of processes where we use grapes or barley or wheat or hops or some other plant-based fermentation process to produce ethanols. So I hope that explains what happens in the process of glycolysis and fermentation. Let me know if you have any questions. Have a good night. Bye.